Hi everyone, my name is Vivian. Welcome to this brief talk on autoimmune disorders of the nervous system. I'm honored to have this chance sharing you our research progress here, and this is my poster. Today I'm going to be talking about multiple sclerosis. I'll begin by outlining the disease's immunopathological role, and then I'll go on to the results we've got so far. Finally, I'll briefly discuss why we feel these new ideas are necessary and beneficial for us. So, I'll begin with the background. MS is a predominantly T-cell mediated autoimmune inflammatory disease of central nervous system (CNS), characterized by multifocal area of immune cell infiltration, demyelination, axonal damage, and disruption of neuronal signal. To this date, some studies have highlighted an important pathogenic role for Th17 cells in autoimmunity, a kind of CD4 T cell subset that secretes cytokine interleukin 17A. However, the treatment for MS still remains a great challenge. Here, we found that small molecule SPAK encoded by STK39 has higher expression in MS patients than healthy control based on NCBI database and can not only suppress the Th17 cell activation but also differentiation. Therefore, we further investigate the underlying mechanism of SPAK in EAE. So, there are many species has a version of EAE, a murine model of MS. In terms of our work, it's in mice, particularly Black 6. Using MOG as the autoantigens, here we have mainly two strategies. We create the T-cell specific SPAK deficient Black 6 mice, then induce with MOG. To further investigate the role of SPAK on T-cell in EAE, we also have the adoptive transfer model. In order to do so, we sorted CD4 T cells from splenocytes of 2D2 transgenic mice, which express MOG-specific TCR, then transfer those sorted CD4 T cells into REC1 knockout mice. Following the strategy, as you can see the timeline here. So, and in figure 1, as I mentioned before, we can see that SPAK encoded gene STK39 was enhanced in MS patients than healthy PBMCs. Simultaneously, SPAK cascade, Wink, OSR1, and NKCC, which is encoded by SLC12A, were relatively higher, as well as TCR signaling related genes such as IL-17A, TNF-alpha, PLC-gamma, FOXP3, and MAPK kinase pathway encoded genes. And in figure 2, we compare the wild-type B6 to SPAK conditional knockout mice. After MOG induction, we analyzed the clinical score, pathological, histopathological, and immunopathological results. And we found that wild-type mice has much severe disease when comparing to SPAK conditional knockout mice. From the histopathological slides, we can see inflammation infiltration at the lateral ventricle in wild-type B6. Furthermore, through flow cytometry analysis, we can see that IL-17A is significantly higher in wild-type immune cells from CNS than SPAK conditional knockout mice. And in figure 3, we use adoptive transfer strategy, as I mentioned in methods before, and clearly we see similar results from figure 2, except the results were more significant. Besides inflammation infiltration, we observed demyelination through looks of fast blue staining. Interestingly, in Figure C, we see that although there were more T cells in cervical lymph nodes from SPAK knockout mice than wild type, in CNS, the number was the opposite. Moreover, through flow cytometry analysis, we found out that T cell activation markers like CD25 were lower in SPAK conditional knockout mice. When comparing these two groups, SPAK deficiency suppressed T cell differentiate into Th17 cells. So to further investigate the role of SPAK cascade in TCR signaling, we use verteporfin, which is an active SPAK-specific inhibitor often used in retinopathy. Through in vitro study, we found out that T cell activation markers were suppressed in a dose-dependent manner. In Figure C, when SPAK is inhibited, the phosphorylation of AKT and its downstream NKCC were also diminished. On the other hand, PLC gamma were increased. In Figure D, we sorted out CD4 T cells from wild type and SPAK knockout mice. The cells were given cross-linking stimulation. And again, phosphorylation of SPAK and its downstream NKCC together with S6K were inhibited, and Z70 were enhanced, which indicated that TCR signaling were affected by SPAK cascade. All of our Western blotting were repeated at least 3 times with similar results. So, according to the above-mentioned results, we can conclude that SPAK inhibits Th17 cell activation and differentiation by affecting TCR signaling, and further protect against experimental autoimmune encephalomyelitis. I'm sorry, I have to stop here due to the time limitation. But if you are interested in our studies, please scan the QR code right here for further information. Thank you.